what I like to do is to discuss nuclear receptors and especially PPARs, and more in the frame of lipid signaling. So there will not be too much about microbiota, not too much about immunology, but I hope that anyway, this signaling will interest you. So fatty acids are really very important signaling molecules in our body. They go into cells. They will bind to fatty acid binding proteins, and these proteins translocate these fatty acids to the cell nucleus, where they bind to nuclear hormone receptors, the peroxisome proliferator activated receptors. So this is a member of this family, which makes a heterodimer with another member of the family, the retinoid X receptor, and regulate gene expression. So these receptors are quite important in metabolic regulation, but also in some basic cellular processes like proliferation and differentiation, and also in cell-cell signaling. And we think that they play a very important role in the general status of our body as we will discuss it. So the general mechanism of action is very similar to other members of this superfamily. So they will bind to specific response element in target genes. They will recruit coactivators, transcription machinery. They will have an impact on chromatin structure, which will lead to chromatin remodeling, and finally stimulate or sometimes also repress gene activity. A very interesting question is for these receptors is the notion of the agonists. I just mentioned to you before fatty acids, but the fatty acids are not the only activators of these receptors. Also eicosanoids, so all sorts of fatty acid derivatives can bind and activate these receptors, which makes them quite fascinating but also more difficult to study because the diversity and level of these molecules will play a very important role. And their level is going to change in function of, I would say, normal physiological changes, exercise, eating, fasting, but also if you have particular pathophysiological situations, inflammation, diabetes, atherosclerosis, cancer injuries. We can even put here enteric infections. Because you will also have a lot of enzymes and which will be involved in making these fatty acid derivatives, lipoxygenases, cyclooxygenases, epoxygenases, lipases, etc., and you have this whole list here of derivatives which are recognized and known as ligands of these receptors. So you can imagine really the complexity within a cell in a specific physiological or pathophysiological situation where you may have many of these ligands which are present simultaneously. So there are three members of this subfamily of PPARs, alpha, beta, also called delta, and gamma. And what I would like to do to give you a kind of overview is to discuss a couple of functions or one function for each of them. So PPAR alpha is well known as a receptor which controls genes which are involved in fatty acid catabolism, and we will see this in a circadian regulated manner. They are important in the control of inflammation, detoxification, and also in sexual dimorphism. So a long time ago already, a PhD student in the lab found out that PPAR alpha in the mouse liver is expressed according to the circadian time frame, if you want. This would be the time point where you put on lights in the animal house, and then you have lights during 12 hours, and the PPAR alpha mRNA and protein is going up, and then when you turn off the light, it's going down again. And liver has indeed a very strong peripheral clock, which is under the control of the central clock. But in this liver clock, other cues play a very important role. For instance, the feeding cycles have quite a strong impact on the clock itself, also rest activity cycles. So this means that if you want to have a good energy balance, it is quite important that anabolic and catabolic processes are coordinated according to these feeding and fasting periods. And the work I am going to discuss now was done by a PhD student, Nicolas Leuenberger, in collaboration with Fred Gachon in Uli Schibler's lab. And what we wanted to know during this cycle of expression of PPAR alpha, are ligands also somehow produced during this time frame? And if they are, what are the mechanisms of the production of these ligands? So Uli Schibler has done quite a lot of work of 3 transcription factors, which are under direct control of the liver clock. So you have here the positive arm, the negative arm of the clock, and you have these 3 factors, which are quite important in regulating several functions. These factors are cycling, similarly to what has been shown before for PPAR alpha. But during microarrays, we found that these factors control these enzymes, these acyl-CoA thioesterases, which are hydrolyzing acyl-CoAs and will liberate 3 fatty acids, which can be ligands for PPAR alpha. They would activate PPAR alpha, and then PPAR alpha target genes will be controlled by the activated PPAR alpha. In this study here, we use these 2 well-defined PPAR alpha target genes to measure PPAR alpha activity. What I have to say also, these 3 factors here have some redundant function, and you really need to knock them out, all 3 of them, to have a full effect on the genes this receptor controls. So these acyl-CoA thioesterases, as I mentioned, are under the control of these factors. They are on the same chromosome, and you have here a target site for this PAR-BZIP transcription factors, which control their activity. And as you can see here, when you do the triple knockout, you have no binding to this site, and in the wild-type situation, you have a very nice circadian binding of this PAR-BZIP transcription factor. You also see that the target genes of this PAR-BZIP, this acyl-CoA thioesterases, show a very nice circadian cycling with a maximal expression at ZT12. And in the triple knockout, you see that you lose this cycling of this enzyme. So does the activity of this enzyme have an effect on fatty acids in the liver? So as you can see here, taking several fatty acids as an example, is that the level of these fatty acids are really also circadian, exactly as the enzymes are, with maybe a difference I am going to discuss in 1 minute, is that you have a second peak here at ZT24. You also see that in the triple knockout animals for these 3 transcription factors, you totally lose the cycling of the level of the fatty acids. And as I mentioned, several of these fatty acids have been described as activators of PPAR alpha, and we think that during the day, the activity of these enzymes are going up. This is under normal diet condition. and they will liberate fatty acids, which will activate PPAR alpha, and PPAR alpha regulates itself and then lead to the control of PPAR alpha target gene. And as you have seen, this is very important at the day-night transition. So we think that PPAR alpha are very high when the animals start to feeding during the night. And probably this whole process prepares the liver metabolically to fulfill its function. So what I would like to mention also is that LPL here, which has a complex regulation, we think, but this is not totally sorted out yet, that it is under the control of this PAR-BZIP transcription factor, to some extent also to PPAR alpha. But as you can see, the regulation of this gene is very different from this thioesteresis. And we think that this lipoprotein lipase, for which the activity is highest at the ZT24, is most likely responsible for the second peak here of high levels of these fatty acids, which would occur at the night-day transition. Now we were interested to know what would happen if we give the animal a fat-free diet in which we will not have fat coming from the diet. And what I am going to show you in this situation, you have a gene which is activated. This is fatty acid synthase, which is producing fatty acids and controls PPAR alpha. In this situation, the activity of PPAR alpha becomes independent of these enzymes. And this is the result. In this fat-free diet, you see that you have a strong stimulation of this fatty acid synthase gene with high level of the mRNA. And you see now that the fatty acids in the liver are not cycling anymore, but are present in sufficient level to activate PPAR alpha. So we see that depending on the availability of fat and ligands, the tissue is using a different strategy. So the first one, with a diet with fat, you will go through this pathway I just described. And if you have no fat, it will go through this fatty acid synthase, which will, this is work of Semenkovich, which described last year that under this situation you have the production of this phospholipid here, which is a very strong nanomolar affinity ligand for PPAR alpha. So this was about the activation of PPAR alpha during the circadian time frame. And what I would like to discuss briefly with you also now is another result which came out from the analysis of this microarrays, in which now we have compared the expression of genes in wild type and knockout animals, and also compared females and males. Again, work done by Nicolas Leuenberger and Sylvain Pradervan for the bioinformatic analysis. So what Nicolas observed is that in the wild type females, he had 3 times more genes which are directly repressed by PPAR alpha compared to males. So this was an interesting observation. What he then found out is that steroid metabolism, cholesterol biosynthesis, and also genes involved in immunity are differentially controlled between male and females, being repressed by PPAR alpha in females. And what Nicolas found out then is that to have this repression in females, you need a stimulation of the ligand binding domain of PPAR alpha, and especially at this position here, which is lysine 358, which, when the receptor is in presence of a ligand, is exposed at the surface of this domain. And if you have an antagonist, in that case, the amino acid would be in the interior somehow of the structure and not accessible to modification enzymes. So indeed, Nicola demonstrated that the ligand, the LBD, needed to be activated to have this stimulation, and this stimulation was then also tissue and also gender specific. What you can see here is that you have here females, the wild type female. Here you have the knockout female, of course, no signal. But here you have the wild type male, and you see this very nice difference in the stimulation of PPAR alpha in this liver tissue. So you have, I mentioned before that genes which are repressed belong for one pathway to cholesterol biosynthesis, and all these enzymes here for which you have this arrow are controlled by PPAR alpha in a negative way. And to find out about the mechanism, Nicola concentrated here on this gene, CYP7B1, which is an enzyme which hydroxylates DHEA and inactivates DHEA. And when the CYP7B1 is repressed in that situation, you will have also in the female liver a higher level of testosterone. And this he has observed very nicely that in the female liver, when PPAR alpha is active to repress this enzyme or the gene encoding this enzyme, you have higher level of plasma testosterone and also liver testosterone. So CYP stimulation is necessary for this repression. What I would like to mention also is that these enzymes is also involved in the metabolism of estrogen receptor antagonists or SERM selective modulators like 27-hydroxycholesterol, which will be important a little bit later. And this enzyme is also involved in bile salt synthesis. So I directly go to the mechanism and explaining you how this CYP7B1 is repressed by stimulated PPAR alpha. So if I take the situation of a PPAR alpha null female, this CYP7B1 promoter is active, and two factors are key here. This GA binding proteins, which has two subunits, and SP1. And these two factors cooperate to stimulate this gene. When PPAR alpha is stimulated, it doesn't bind to the promoter of this gene, but it binds directly through sites we have identified to this GA binding protein subunit alpha, and then it recruits repressor, histone deacetylases, and also we don't know exactly the sequence of events in details. It also leads to removal of SP1, which is then methylated. So you have this CpG, the cytosine is methylated, and this leads to a downregulation of this promoter, which also changes here in the chromatin structure. So this interaction of stimulated PPAR alpha with this subunit of GA binding proteins leads to a repressor complex, which is somehow a novel mechanism of repression of genes by PPARs and nuclear receptors, which has not been described so far. So what could be really the signification? So now I enter a little bit in a speculative mode. So what we know is that, as I showed you, PPAR alpha in female represses CYP7B1. I also showed you that when CYP7B1 is repressed, you will have higher level of androgens, which themselves also have a negative effect on the expression of these genes. And we think that this somehow counteracts the effect of the estrogen receptor, which indirectly stimulates this enzyme, which, as I mentioned before, will clear estrogen receptor antagonist. And this, in absence of this negative mechanism here, may lead to an excessive action of the estrogen receptor and its activity, which can lead to hepatotoxicity and cholestasis. So PPAR alpha would protect against estrogen-induced hepatotoxicity, and this we have really tested in females, and it works very nicely. One can reduce this hepatotoxicity if one treats this female with a PPAR alpha agonist. So PPAR alpha is also the target of hypolipidemic drugs, which are the fibrates, and if you treat these mice with fibrates, you can reduce hepatotoxicity after a very strong estrogen stimulation. So does this have a significance for medicine? We don't know exactly, but what is known is that in situations of very high level of estrogen, you can have this intrahepatic cholestasis of pregnancy, which incidence varies a lot in different populations. It goes from 1 or a few percent to up to 15%. This estrogen-induced hepatotoxicity has also been observed, for instance, in susceptible women taking estrogen-containing oral contraceptive. The point is that there is no really good drug so far to treat this intrahepatic cholestasis of pregnancy. What is used in general is ursodeoxycholic acid. And what we would like to propose, nobody has done the experiment so far, is that maybe PPAR activators would somehow help to treat this woman. This is totally speculative. Now let's move to PPAR beta. And here I would like also to discuss fatty acid metabolism, catabolism, taking as an example skeletal muscle. This is work we have done in collaboration with Daniel Metzger and Pierre Chambon. So what we did here is to generate mice in which PPAR beta is specifically deleted in muscle tissue, in skeletal muscle, and we did two types of construct in which in one construct we had the constitutive deletion in muscle, and in another construct we could induce the deletion in the adult. Basically the results were the same. So what did we observe? In animal in which PPAR beta is deleted only in skeletal muscle, we see that we have a change in the structure of the muscle. We lose oxidative fibers, which are here in dark blue, and here you have this conditional knockout. This loss of oxidative fibers is paralleled by a reduction of mitochondrial electron chain, electron transport chain, and also a reduction of fatty acid catabolism and energy uncoupling, as you can see here. So in terms of muscle physiology, quite an important effect. And indeed now, if we look at the long-term effect of this mutation in skeletal muscle, when you have the animals under a normal diet for about 25 weeks or a little bit more here in this experiment, you can see that the knockout animals become obese compared to the control diet animals, as you can see here after 25 weeks. When you look at the adipose tissue, you can also see here that the cells are much bigger than in the control animals, and the fat content is increased dramatically. So if I summarize what we have observed, is that oxidative capacity is reduced, fatty acid uptake and binding and breakdown is reduced in these knockout animals, expression and activity of mitochondrial enzymes is decreased, and also very interestingly, the performance of these animals is dramatically reduced. What we observed also in this animal, when they age, they will develop obesity and then insulin resistance. And we think that these experiments tells us quite important things, because it has been described since quite a bit of time that we know that in obesity and diabetes, you have in general a loss of oxidative activity in muscle, but what was not known was if it was the cause of the consequence. And I think that our experiments show that if you reduce the oxidative capacity of the muscle in a healthy animal, with time this animal will develop obesity and diabetes. So what is the molecular mechanism behind it? We think that PGC1 alpha, which is a coactivator of the PPAR beta receptor and also the other PPARs, plays a major role. And what is interesting is that this PGC1 alpha encoding genes is a target gene of PPAR beta in muscle. So what is occurring in muscle when PPAR beta is activated, it will stimulate the expression of PGC1 alpha, which will somehow stimulate the activity of the PPAR beta receptor, but also of other PPAR beta target genes. But PGC1 is also a coactivator for other transcription factors like NRF, which is involved in the expression of nuclear encoded mitochondrial genes, and also of MEF2, which is involved in the control of genes which are involved in the fiber twitch in muscle. So I think this is quite an important mechanism which explains, at least in part, the effect we have seen on these knockout animals. To finish, I would like to move now to PPAR gamma and discuss a little bit inflammation. So all three receptors are involved in inflammation control, and really the first link between PPAR and inflammation was done by one of my postdocs years ago, Pallavi Devchan, which came up with a model where she postulated that activated PPAR alpha in liver would somehow induce the catabolism of inflammatory eicosanoids like LTB4. This has since been confirmed. So this is one of the mechanisms by which PPARs control inflammation. But most of the time, PPARs are involved in negative regulation of gene expression, acute phase response genes, especially also inflammatory cytokines. And I would just like to illustrate a little bit more PPAR gamma in the gut. So in epithelial cells, PPAR gamma and in the gut in general is quite important to control inflammation. And the first work in this field we had done in collaboration with Pierre Desremaux years ago also, when we got the PPAR gamma heterozygous animal at that time, because full knockout is lethal because of placental defect. We showed that this PPAR is quite important in the control of inflammation in the gut. Now we know a little bit more through the work of many people. This is certainly not only our work, that PPAR gamma, when activated, especially by omega-3 fatty acids, for instance, but also by synthetic molecules. But if we take a kind of more natural situation, when we have high levels of omega-3 fatty acids, you will have activation of this PPAR, and you will counteract here the inflammatory pathway to reduce the production of cytokines, adhesion molecules, and reduce inflammation. What is interesting also is that the regulation of the activity of PPAR gamma in these cells is quite complex. It is not only activation by ligands, omega-3 fatty acid, for instance, but also the microbiota has an effect on the phosphorylation of PPAR gamma. This was shown by Swen and his collaborator, and modulates the activity of PPAR gamma. It has also been shown that LPS itself modulates the expression of PPAR gamma by mechanisms which are not quite solved at the molecular level so far. And omega-3 fatty acids can also have an anti-inflammatory effect, which are independent of PPAR gamma. This has been shown recently in macrophages by Olewski, where this omega-3 fatty acid binds to this membrane receptor, and then through this pathway here, again interact with NF-kappa B and the P1 signaling. PPAR gamma can also, as again demonstrated by Swen and collaborators, have an effect on the nuclear localization of NF-kappa B subunits. So you see, this brings me to the end, and I will come back to the discussion a little bit about tornado, because we would really like to use now all these animals models we have now to really study and go in the direction of what has been discussed during these 2 days. How are these receptors involved in immune responses? How are these receptors involved in microbiota signals? So what is somehow interesting with these receptors is that it's a little bit like microbiota. Okay? At first sight, one has the impression that they are involved in everything one looks at. It is true to some extent, and for sure they are involved everywhere where you have lipid signaling. And I discussed today PPAR alpha in liver, PPAR beta in muscle, gamma in the intestine. But for instance, PPAR beta is also involved in insulin secretion. This is ongoing work. We're using, again, tissue-specific knockout. And very early on in development, and this is quite interesting, before even cells start to migrate in the embryo, we know now that PPAR beta controls the very early step of gastrulation, when the cells in the early embryo lose pluripotency. And also in tissue repair, and especially PPAR beta in the skin. We use a model here of skin cancer. Now, I would like to, of course, acknowledge the different people, but what I would like to say and repeat myself, I think in this very different situation, in diabetes, in muscle, in the gut, in liver, I think it will be quite interesting now to see how these PPARs are involved in microbiota signaling in different organs of the body. And just to finish, the different people who have been involved: Nicolas Leuenberg in the liver project, Pallavi Devshan in the inflammation, Aubry Tardivel, Beatrice Devergne in the muscle project, bioinformatics by Sylvain, and to get these very different genetic models and experiments, collaboration with Daniel Metzger, Pierre Chambon, with Denis Duboul for the PPAR beta knockouts animal, and Uli Schibler and Fred Gachon I mentioned before. So thank you very much for your attention.